degraded, and many other parts of the cell are being degraded by autophagy to provide amino acids, and either to be used as an energy source or to be remade into some other protein. So the fact that there's more autophagy going on in mice that eat lean and don't eat enough calories seems to be protective, because if you have a mouse that cannot do autophagy or have some aspect of autophagy knocked out, they don't have that protection. And I think they've done this in human cells, that they've taken it into human cells. So autophagy is important. You can't ignore it anymore.
Dr. Singer, I touched a mouse that eats more will probably produce excess protein, which causes autophagy to happen. How do mouse that eat lean diet do more autophagy than the ones that?
The need for energy or amino acids will stimulate the autophagy of proteins in the cytoplasm.
So is it like replicating like amino acid starvation?
You could probably trigger autophagy by that mechanism. Not that proteins are unimportant, but the cell will find multiple ways of getting energy and amino acids. Okay, what we need to do is look at how does a lysosome that is made, a lysosomal protein gets made in the ER, how does it get sorted in the trans-Golgi network to be delivered either to an early endosome or a late endosome, okay? And some will accidentally escape them. Yeah, Amanda.
Why doesn't it just develop straight into a lysosome? Like, why do you need the early, late, and then lysosome steps?
I don't know if it's just simply a maturation thing, which is kind of what I'm thinking, or this is another possibility. Is the cargo, the protein that's going to take the cargo to the late endosome or to the early endosome has to be recycled so it can be used again. Maybe if it went straight to the lysosome, it would never make it back to the trans-Golgi network because it would be degraded. It could be something that simple. Briles?
So in the diagram that you have, it shows like the TGN creating the early endosome. It's-
Okay, got it.
I know you did the outside of the cytoplasm stuff.
Uh-huh.
Is the thing that shows the budding off, is that the TGN?
Oh, on the early endosome?
The just that part right there.
Yeah.
Like this, is that part of the trans-Golgi network or not?
No. So what they're not, this is only showing internal. So what's going on here? See this
So it's
this arrow?
this part.
Yeah.
Okay.
So this arrow that's it has something from the cell surface going to the endosome, early endosome. That's that.
Okay.
That's going to be receptor-mediated endocytosis. It should have a little red coat around it. And I should never keep up with this stuff because I've already found out that I have to change what I'm going to talk about when we get to the trans-Golgi network. Okay, so up in the cis-Golgi, lysosomal proteins will be modified. Okay, so we're going to start out with a protein that happens to be in the cis-Golgi. Okay. Lysosomal proteins have to be glycosylated. Okay, you remember N-acetylglucosamine, mannose, by the time they get to the, remember, by the time they get into the cis Golgi, that mannose is taken off. Okay, do you remember that?
Yeah.
Okay, so this is what is received by the cis Golgi. So in the cis Golgi, whoops. Okay, we're going to modify, and I should show you the process. Oh, man, that's not simple. Okay, this is the same protein, by the way. A phosphate group is added to that particular mannose. And now this is then, this lysosomal, this is going to be called a mannose 6-phosphate.
Minus 6 or minus 8?
Okay, so a mannose 6-phosphate is created on the oligosaccharide of lysosomal bound proteins. Okay, here's the name of the protein. You'd probably need a whole page to write the name of the protein. And then I'm going to show you why it's called that. It's the strangest reaction. Don't you think a nice old ATP hydrolysis would do the job? But no. I'll show you how complicated it is. Here's the name of the enzyme. N-acetyl glucosamine. Phosphotransferase. Okay. And what does this? This is the enzyme that has to recognize what is a lysosomal protein and what is not a lysosomal protein. Okay, because it'll only add phosphate onto that mannose for lysosomal proteins. We'll talk about that in a second. Okay. All right. See if anybody recognizes this molecule? Okay, does that, anybody recognize that? Okay. You recognize it? It's a ribose, yeah, it's a ribose with uracil. So this is called a uridine, monouridine. Okay, I just drew it backwards. I normally put the 1' carbon on the other side, but I drew it backwards. Okay, good, you got that part. Oopsie. Okay. Okay. That sounds like a vortexer. Slow one, but a vortexer. Okay, so this is going to be the source of the phosphate. We're not using ATP, This is the source of the phosphate, it's uridine diphosphate. N-acetylglucosamine and uridine diphosphate. So here's the N-acetylglucosamine. And uridine diphosphate. Or UDP. So this would be your UDP and acetylglucosamine. Okay? So I'm going to abbreviate it this way, so because this is getting to be ridiculous. Uridine, okay? So this part is here. Two phosphates, and acetylglucosamine. Okay. I'll just do that. That looks like the lysine. Okay. Got that? Oh, I didn't finish this up. Aha, yes I did. Okay, so this gets transferred. I'm just gonna draw the okay, there's the mannoses. The N-acetylglucosamine is there, and the block with the N-acetylglucosamine. Okay, so we're going to split the bond and attach the phosphate and the N-acetylglucosamine to the mannose, and then we're going to cut the N-acetylglucosamine off. One of the cells more ridiculous. Whoops. Close enough. That's one of the cell's more ridiculous reactions. So you add, you start out with the uridine UDP N-acetylglucosamine. Inside of that UDP N-acetylglucosamine is the phosphate that is eventually going to be transferred to the mannose. So you put this part onto you put the one phosphate and the N-acetylglucosamine onto the mannose, then you cut the N-acetylglucosamine off. And I think it would have been a whole lot easier if you had just done ATP, used ATP.
Is there supposed to be only 7?
Oh, no, there should be 8.
8, okay.
I'm missing one. Oh, yeah, you know where the one is,
Yeah.
is probably that one right there. Yep, it's this one right there. That should be a circle. I way abbreviated it. Isn't that weird? I just can't figure it out.
What
Yeah.
cuts off N-acetylglucosamine?
Oh, it's the same enzyme. It's the same enzyme. It's a multi-step enzyme. Okay?
So it's that phosphotransferase deal.
Yeah, so N-acetylglucosamine phospho, that's what's getting transferred, okay, from UDP to the mannose, okay? So N-acetylglucosamine phosphor, or phospho N-acetylglucosamine is being transferred from UDP to the mannose, and then the N-acetylglucosamine just gets cut off by the same enzyme, Okay, but the source of the phosphate is the UDP N-acetylglucosamine.
So it gets cut off by the N-acetylglucosamine.
Yeah.
phos-
Yeah, it does this whole stuff.
Okay.
And
It would have been a whole lot better for it to use a kinase, but.
And you said that enzyme is what can tell that it's
Yes.
supposed to be a lysosomal protein?
Yes, and the way it tells is not by a particular amino acid sequence, but by a particular conformation on the protein. Okay, so there must be, let's say, maybe there's a bump here, let's say maybe this is the book has a kind of an interesting, they do something like make a little finger that's kind of unique to lysosomal proteins. Okay, what that means, the reason why it's not an amino acid sequence is there's a variety of ways to generate that particular conformation. All you have to do, all the lysosomal protein has to do when it folds is create that conformation. And there are really no amino acid rules. And the other thing that's probably true about this conformation is that they're not contiguous amino acids. In other words, they're not just a string of amino acids, but they're amino acids from other parts of the protein. Okay, this is what's generally called a signal path. Okay, and this is a conformation. So a particular conformation can be a signal patch. And for N-acetylglucosamine phosphotransferase, it is looking for a specific patch or conformation on proteins, and that identifies those proteins as a lysosomal protein. Okay, does that make sense? So no contiguity, a little stretch of amino acids is a shape that the N-acetylglucosamine phosphotransferase is looking for. And this shape has to be unique to lysosomal enzymes.
So do signal patches like that require a specific like protein folding chaperone, or does it?
You know, I don't know. I'm guessing no. I'm guessing no. Because if that were the case, then there would, what would happen if that protein was mutant, that specific protein folding chaperone was mutant?
Then you wouldn't have the.
You wouldn't have any, you wouldn't have any functional lysosomes. Okay.
Why is it called patch when it's more of a confirmation?
What?
Why is it called patch?
Patch? I think patch for region, it could be domain. I don't know why it got called patch, but it could be, I would call it a signal domain, but, okay, so a piece of the protein. So maybe that's where the patch came, a little patch on the protein.
So the dent is what it's referring to, the dent in the protein.
Right. Yes. So once this phosphate has been put on this mannose, okay, the protein travels all the way through to the trans-Golgi unmodified. This is the last modification that this protein will pick up in the Golgi. Okay, so it'll make it into the trans-Golgi network with no more modifications. What do you suppose is going to happen to proteins if N-acetylglucosamine phosphotransferase was not present in the cis-Golgi? Let's say it was mutant. No, if it was completely missing from the cell, what would happen to lysosomal proteins?
Goose can
It wouldn't make it to the lysosome where they end up. Does the bulk flow pathway, if that address
goes out of the
label is
cell.
not there,
They'll go out the cell.
they'll be secreted, okay? So if N-acetylglucosamine is not functional in the cell, then these guys are gonna take the bulk flow pathway, because this phosphate bumps them off the bulk flow pathway. Okay? And if that phosphate doesn't get put on in this crazy way, this guy comes off, this guy's gonna be secreted by the bulk flow pathway. What's gonna happen to the lysosomes?
They're not gonna exist.
Yeah, there's gonna be no proteins in the lysosome, okay? And there is a disease with mutant N-acetylglucosamine phosphotransferase. I'm just gonna call this mutant enzyme, but it's this enzyme. Okay, so this enzyme is mutant. The person has inherited the disease I-cell disease. Okay, I for inclusion. And an inclusion is a very densely stained body in the cell. Here I use the word body, but I don't mean human. So what do you suppose that densely stained entity in the cell is? You can, something is extremely visible in people with I-cell disease.
Would it normally not be visible?
Huh?
Would it normally not be visible?
Right, yeah. Yeah, we hope you, I can assure you, you don't have these. Because you're sitting here today, getting ready for a test.
Maybe I'll have it tomorrow.
It might happen tomorrow. The inclusion bodies are lysosomes because notice that all of the stuff coming into the lysosomes, stuff from receptor-mediated endocytosis, stuff that gets autophagocytized, goes to the lysosome. Okay, they're not getting degraded because there aren't any hydrolytic enzymes or degrading enzymes in the lysosomes. So the lysosome becomes bigger and bigger and bigger, and it's chock full of undegraded stuff. This is not so bad really for cells that divide, like your intestinal cells, because because that great big lysosome thing is going to get broken into smaller fragments. But where do you suppose that might cause a problem?
Brain.
The brain. And so this is a neurodegenerative disease. It's also treatable. That reminds me of a Harrison Ford movie. The name isn't counterintuitive to me, so I'll have to find the name. Okay. So I-cell, this address label is obviously important. Okay, I-cell disease is different from, a little bit different from lysosomal storage diseases like Niemann-Pick. And the reason is, in other lysosomal storage diseases, one enzyme, an enzyme that should function in the lysosome, is mutant. The mutant protein gets to the lysosome, but it's not functional. This is one where nothing gets to the lysosome. And people don't live very long. Okay, so when we so on Monday we'll pick up with the Trans-Golgi network and then look at, because we still have to get the lysosomal proteins to the early endosome or late endosome. So we'll look at that next.